Hi, everyone. Welcome to NC State's Wicked Problems Wolfpack Solutions tonight. I am Dr. Bucky Gates. I'm a faculty member in the Department of Biological Sciences, and I'm a paleontologist. Most of my work, I focus on fossil sharks, I focus on dinosaurs and ecosystems from the very end of the Cretaceous period. And so tonight we have a very special guest and a really great opportunity. We're talking to Dr. Lindsay Zanno, and she is in the middle of the Utah desert. And so what we're going to be doing tonight is we're going to be talking to her at a really special site that she has been digging for many, many years now. And so I hope all of you got to see her video previously about her work in the Middle Cretaceous. It is a really great video, and if you haven't, please catch up with that one. But what we're going to be doing tonight is having her extend on some of that stuff. And so she's going to be talking about a mass death assemblage called dinosaurs, drought, and disease. And so with that, I want to go ahead and introduce Dr. Zanno. She is a faculty member of the Department of Biological Sciences as well. She's also the head of paleontology at the North Carolina Museum of Natural Sciences in downtown Raleigh. So Lindsay, hi.
Hey, Bucky, how's it going?
Oh, it's going great, thank you. So I imagine that the weather here, with us having cool weather with a tropical depression coming through today, thank you, Elsa, is very different from what you guys are experiencing with 110 degrees in Green River, Utah.
Yeah, it's a little bit toasty out here today. It's extremely hot and extremely dry. The Southwest is in a record drought and a record heat wave, which is a great combination if you're out digging dinosaurs in the sun in the middle of the desert all day.
That's perfect. And it seems like you constructed that segue absolutely for me, because what I want to do is go straight into the site that you're talking about. Why in the world did you want to title your talk Drought, Dinosaurs, and Disease?
Well, this is an interesting dinosaur quarry that we're at. It's a very rare quarry, not only for the time period that it represents, it's very early in the early Cretaceous period. So this quarry is somewhere on the order of 137 million years old or so. And that's a time in dinosaur evolution where we don't have a lot of dinosaur fossils from the North American continent. So it's a very rare ecosystem that isn't preserved in a lot of places around North America. So we're lucky to have a dinosaur quarry here. And that's one thing that makes this dinosaur quarry so special, is it fills in this gap in this time period. But the other thing is that it's an unusual quarry in that we have this entire hillside and it's filled with a mass death assemblage of the same species of dinosaur. The species of dinosaur we call Falcarius utahensis. And this is also a very rare type of dinosaur, but we happen to have somewhere in the order of an estimated 300 individuals or so buried at this site, including small babies all the way up to like 4 meter, 5 meter adults.
Wow, that sounds amazing. So there's two things I want to get to first. So I want to talk about the dinosaur itself, but also this deposit. So first, we'll talk about the dinosaur, and then we're going to kind of move to this deposit. I'm going to share my screen for just a second to show people what Falcarius looks like based on illustrations from your previous work. At least I think I am. Yeah, here we go. All right, so this is a skeleton. Everyone is looking at a skeleton at the moment for Falcarius. And then I'm going to switch over to the fleshed out reconstruction. Can you just tell us a little bit about what this dinosaur is?
Yeah, well, therosaurs are really fascinating dinosaurs. They're members of the theropod dinosaur group, which we colloquially think of as predatory dinosaurs, and all the predatory dinosaurs are part of this group. But therosaurs actually were plant-eating dinosaurs. They were part of the dinosaur group most closely related to birds, so they're pretty close to the bird split. And so They have, larger brains than a lot of other dinosaurs and good sensory systems and a lot of bird-like characteristics. But early on in their evolution, they sort of took a turn from the predatory role and became plant eaters. And they're pretty much extremely rare around the world. We only find their fossils definitively in the northern land masses, Asia, Europe, and North America. And there's only 3 species known from all of North America so far.
Oh, wow, great. And so you're working at one of the sites. So of the 3 species that are currently known, how does this relate to the bigger picture of therizinosaur evolution?
Well, the species that comes from this quarry is actually the most primitive therizinosaur that's yet been discovered. So when it was described initially, it provided us sort of a quote unquote missing link. That is a really transitional animal that helps us to pin down where this group belongs on the family tree. So these end up being really important species because as a lineage evolves and becomes more and more specialized, it can be really hard to link it to its ancestry. So you really need to find those animals that allow you to reconnect those steps so you can trace back what the ancestor would have looked like. And Valkyrius really helped us do that when it was described about 16 years ago.
Okay, cool. So the reconstruction that I have up right now shows about the size of Valkyrius as well as feathers on it. Do you really think it had feathers?
Absolutely. All of the relatives we have that are well preserved, that is in a depositional environment that allows us to see what kind of skin or integument they had feathers. So even though we don't have feathers at this quarry, we know if their ancestors and their descendants all had feathers, then this species undoubtedly had feathers as well.
Okay. And so you don't have feathers preserved in the bones at the quarry itself, right?
That's correct.
Okay. So what is preserved at this site that you're sitting in?
Well, we have hundreds of bones per square meter. The bones are extremely fragile. One of the features of theropod dinosaurs is they have very thin, lightweight bones, with a lot of air spaces, and so the bones break apart very easily. That's one of the things that makes theropod dinosaurs so hard to find in the fossil record is they're not easy to preserve. So we have to go very, very carefully in this quarry so that we don't damage the bones because they're very thin and small.
Okay. And so how many individuals do you think you have? 1, 2, 3,000?
Well, it's really tough to say because we're not really sure. This is an enormous hill, but we've sort of made a back of the envelope calculation and there are at least several hundred individuals buried here.
Okay. Well, your camera went off, Lindsay. I don't know if you have some camera issues and not being out in the middle of nowhere. Oh, there we go. Excellent. Great. Okay, so what about other fossil sites in the American West? Are there other sites that have lots of dinosaurs, or is this a really unique one?
Well, sure. There are plenty of sites that have many individuals buried. They tend to be pretty rare for theropod dinosaurs because theropod dinosaurs themselves are very rare.
Mhm.
They're, typically at the top of the food chain, so their population numbers in the ancient ecosystems would have been lower than herds of prey, right, that would have roamed those ecosystems. So right from the get-go, there are fewer of them to get preserved, which makes it harder to find them. And then it's going to be harder to find them in mass. But of course, we think Valkyrius was a plant eater, so its ecology may have been very different. It may have lived in herds or lived in large numbers of individuals. And we're still, even though this quarry has been worked for many, many, many years now, and there have been many publications, we're still trying to figure out exactly what happened that brought all these individuals together, that killed them, that accumulated all their bones in one place. So it's a very complicated story.
Okay. Well, it sounds like complicated work to go along with a complicated story. And with so many individuals in there, so many bones. I'm really curious about what kind of processes, what methods do you use to make sure that bones from this quarry stay with this quarry? What things do you do in that quarry there to make sure you can reassociate one bone that was next to another one once you get it back into North Carolina?
Well, one of the jobs of a paleontologist is not simply to discover something and dig it up and remove it. We're collecting a lot of data from the quarry, right? We want to know the three-dimensional orientation of all the bones, how they relate to one another, so we construct maps so that their original position relative to one another is preserved. Unfortunately, at this quarry, the individual skeletons decomposed all the way before these animals ultimately got buried at this site. So the bones are completely scattered, although there does seem to be some differentiation between the bottom of the quarry and the top, where we find extra lightweight bones higher in the quarry. So maybe that as the water was moving in, those bones settled out last because they were the lightest. So we see some patterns that we're beginning to decode across the quarry, but unfortunately that means if we find a toe bone and then a meter away we find an upper arm bone, we can't say that those belong to the same individual just because they were close together. It seems like the individuals were all jumbled up together before they were buried.
Okay, it sounds like a really difficult kind of a prehistoric forensics case or something, like a CSI kind of a thing.
It is. That's what makes it so fun. Is this is not a simple puzzle to figure out. It's going to take many, many years, and we have started turning up a lot of bones from a sauropod dinosaur in this quarry as well. We have some almost meter-sized vertebrae from the site, and so that's a animal that may or may not be new, maybe a new species, but we're just sort of beginning to collect the bones of that animal, so maybe we can figure out what kind of sauropod or long-necked dinosaur species that actually is.
Wow, really fascinating. Okay, so you see a lot of people behind you. What's happening at your quarry now? Who's there?
Yeah, so the grad students from my lab are here. So those are students doing their master's and Ph.D. degrees in paleontology right now, want to go on to have their own research labs. And we also have a team of undergraduates from NC State out right now. Every other summer, I teach a course called Bio 325, which is paleontological field methods. And we do a lot of hands-on training in the lab as far as like how to handle and conserve fossils and what kinds of data we collect from the field, and a little bit of museum conservation work. And then we bring the students out into the field and they can have a couple weeks of hands-on experience excavating fossils here in the quarry.
Oh, that's great. So what about other opportunities at NC State for paleo?
So NC State has a brand new paleobiology minor for students who are interested in learning more about what paleontology is all about. And it's a great way to, if you think you might be interested in research experiences in paleontology or fieldwork experiences, getting into the minor and taking a few classes and seeing if paleontology is something that sparks your interest is a great way to go.
Okay. Well, it's almost time to get questions from the audience. But before we do that, I do want to ask about a little bit of dangerous stuff. So, obviously, having 110-degree heat is dangerous things. I was looking at your Twitter feed beforehand, and you had rattlesnakes out in a previous site. I hate snakes. I'm really glad I wasn't there. But I'm curious, what kind of dangers do you see at this site?
Well, these are pretty mundane dangers, but the heat can be life-threatening. So we have to take care to try and put up shade and keep an eye on everyone, make sure everyone stays hydrated. I did see a baby rattlesnake by my tent this morning. We have horny toads out there, and I did get stung by a scorpion in the quarry this morning. So that wasn't very much fun, but these are all things that come with the job.
Stung by a scorpion and you're not dead. That's good.
Yeah.
I thought scorpions were deadly. All right, well, let's go ahead and start taking some questions because they're really pouring in, and some of these are just spot on. All right, so since you know that the course is kind of about like kind of large scale global change, I want to take some that get to kind of the big picture of what you're doing in the mid-Cretaceous. There was a great question that I can't even begin to answer, so I'm glad that you're here. And where did this go? Aha, here we go. Viviana wanted to know, based on the information that you have already collected, did you notice any similarities between the organisms that survived the mid-Cretaceous period? And if you did, what did they have in common?
Well, I mean, this is a ongoing research question, so I hesitate to really say too much about it because I think this is the aim of our study, and it's going to take several years to get there. But it is interesting, just sort of grossly looking at it without any really sophistication. It seems to be two patterns, which is that a lot of the dinosaurs that were living in North America, these more sort of archaic or lineages that were already here leading up until the time period when we saw this great deal of climate change, actually didn't really survive that change, right? And the lineages that did survive seem to have Asian ties. So these are animals that probably were living in Asia. Maybe they were better adapted to these conditions. We don't really know, but they migrated across the Alaskan land bridge into Western North America, and they seem to have done really well after most of the North American dinosaurs went extinct. And I kind of want to draw, maybe an important line there, because oftentimes when people think about what goes extinct versus what succeeds, they sort of think there's something inherently better about the animals that succeed. And I think that's the wrong way to look at it. Really, those animals were just doing fine the way they were. But when the animals that were living here in North America that were already occupying all those ecological niches succumbed to the climate change, in other words, they just couldn't keep up with what was happening here in North America, that left all these opportunities vacant. And so those Asian dinosaurs that were here were able to take advantage of that, but it doesn't mean they were just out competing the animals that were already living here, right? If we didn't have that climate change and if they didn't succumb to that extinction, those lineages probably would have gone on and done just fine for many more millions of years.
Okay. Well, that's actually really cool because Sarah asks a question that is quite prescient with what you just said. And Sarah says, what do you believe the results of modern human-induced climate change will have on our planet in the next 300 years versus the gradual change that occurred during the Cretaceous period? Do you predict much more devastating results if major changes are not implemented soon?
Yeah, absolutely. I mean, this is what humanity's dealing with, right? We know that what the changes that occurred in terms of rising atmospheric gases and global temperature rise that happened in the mid-Cretaceous are within what's projected to happen to us in about 300 years. But the difference really is the rate, right? These changes that happened in the mid-Cretaceous, they happened over tens of millions of years, right? In some cases, maybe a few 100,000 years for particular spikes in temperature. But the rate at which we're experiencing change right now is so devastating because animals don't have time to evolve to keep up with it, right? So it takes time for lineages to adapt. And usually what we see with the changes happening right now is we see populations moving. They're sort of tracking their environments. Maybe they're moving up elevation to stay at the right temperature, but eventually, you get to the top of the mountain. So the real issue with what we're going through now in our current crisis is the rate at which it's happening. And we are actually already in a sixth mass extinction event that we have caused through human-induced climate change. So whereas there was a major extinction event in the mid-Cretaceous, it isn't a mass extinction. It isn't like 3/4 or 4/5 or 5/6 of all life on Earth went extinct during that time. It was more of a stepwise change where some lineages went extinct and other lineages evolved. And so it's kind of a more gradual shift.
Okay. And so it sounds like, given that we know the rate of change today is happening a lot faster than lineages can evolve, it seems to me like paleontology has an important role to play in understanding how ecosystems could possibly respond. Is that right?
I mean, absolutely. You can't think about, what's going to happen in the future when you have all this data that paleontology can provide about what's the baseline of what's happened in the past, right? These are real experiments, if you will, that have already taken place on Earth, and that those data exist. We just need to kind of dig them up and figure it all out. But we wouldn't know what a mass extinction is. We wouldn't know whether the kinds of rates of change we're experiencing now were normal or not if we didn't have the paleontological and the ancient geological data of Earth's history to compare them to. So those data are essential for putting what we're experiencing now in perspective of the 4 billion year history of life on Earth.
Wow, cool. Well, I want to thank Tia for asking that question. It seems to be just spot on. All right, so now that we've covered some really kind of downer stuff about what's coming up in our future, let's go back in time, talk about the dinosaur where you're sitting in, the dinosaur quarry. Rohan wants to know about the biome. So what was the environment that was around?
Yeah, so this was a pretty dry, arid environment at this time when these animals were around. The quarry itself seems to be somewhat of a natural spring where we had some sort of fresh spring water here. And, the reason we titled the talk what we did is there are sort of two hypotheses for what might have happened to these animals. One being, a drought, and then some mass accumulation of those skeletons. The other potentially being a diseased waterhole because we know that there's evidence for a spring here. So we have sort of a dry, arid landscape at this time in Western North America with sort of these spring environments around.
Okay. So, for me, it seems really hard to understand how you could possibly determine if a disease killed all these animals. Because I can't imagine a virus or a bacteria preserving in the fossil record. It just seems like such a rare thing to happen. What kind of stuff would you like to see?
Microbes can preserve in the fossil record and do preserve in the fossil record, but no, we don't have any evidence at all that there was, a particular disease, and that doesn't usually show up. Those kinds of things don't usually show up in the skeleton. So it can be really hard to diagnose those things, which is why it's one of the hypotheses that's still out there because we can't disprove it, which is, the way that we go through these things is we have a set of hypotheses and we try and disprove ones until only one is remaining. And we can't rule out that there was some kind of, toxic environment here in the spring that could have killed animals and they could have accumulated here. Nor can we really rule out that there was a drought or many other various hypotheses for how these animals got assembled in this one place.
Okay. So it really is like you're testing things, the legit scientific method. You're not just out digging and waving your arms about what would have happened. You make legitimate hypotheses, you label the data that you need to support one versus the other. You collect the data, you see what supported at the end of the day. And it sounds like it's possible that multiple things might be supported because you don't have enough data. Yeah.
Yeah, absolutely. That's, one of the things that happens with paleontology is there's a lot of filters that, we can't run the experiment to a T exactly the way we want because it's already been run, and our job is to kind of collect what data remains and figure that out. We do have a student in my lab, Kyla Begues, who's working on paleopathology, and she's doing some sectioning of some diseased bones from Valkyrias to look at, what kinds of disease processes might have these animals have suffered before their death. But that doesn't necessarily mean that's what caused their death. It could have just been things they suffered while they were alive. On the other hand, if there's some major percentage of animal skeletons in here that have a particular disease that's evident on the skeleton, that might actually give us some information.
Okay. And so Valerie wants to know, do you ever study animals that are alive now to gain a better understanding of your dinosaur finds?
Absolutely. That's actually precisely what we do. We study both the dinosaurs that are alive today, which are known as birds, which, are incredibly valuable for helping us understand dinosaur biology. And then we bracket that family tree by studying the other closest living cousins to dinosaurs, which are crocodiles. And so usually we will look at the biology of crocodiles and birds, and we'll use those to make inferences about dinosaurs because they sort of fall, extinct dinosaurs sort of fall right in the middle. Unfortunately, crocodiles and birds are very, very, very different animals, at least modern ones.
Mm-hmm.
So it's very rare to find any aspect of the biology that they share that we can infer really confidently all dinosaurs would have shared too. And so it becomes a little more nuanced when we start diving into dinosaurs.
Okay, this gets to kind of an interesting point. So based on my experience, I know that both birds and crocodiles survived that KT extinction. Big asteroid comes down, blows apart the worlds, all the dinosaurs that are not birds go extinct. One lineage of birds does indeed go extinct. And yet somehow crocodiles and modern day birds make it through. You just said that they're so different. Can you give us some idea, some hypothesis as to why you think that crocodiles and birds both made it through?
Well, look, there are a lot of people that work on this question, and there are a lot of ideas out there. Some things we know about surviving mass extinction events is having a widely distributed population. There are very few key indicators in biology that are linked to survival and mass extinction. Some of those things potentially being burrowing or potentially being globally widespread, because if you're everywhere, it's likely some part of your population is going to survive. Something we know about the KT is a lot of things that had links to water survived, especially freshwater environments. So, maybe that water helped mitigate some of those devastating effects for crocodiles. And it may be that for birds, being able to migrate or move across large swaths of area helps them. But it's very, very, these are legitimate hypotheses that are supported with various lines of data from different research groups. But I don't think we really have ever come to the final say on why certain things survived the KT extinction and certain things didn't.
Okay. Well, you're doing a lot of great work in the mid-Cretaceous, and you say that there's still a lot of work to be done on the KT extinction. Is there any kind of research project you're doing to help with that KT extinction research?
Yeah, we are working in one of the youngest extinct dinosaur-bearing formations in North America, the Hell Creek Formation up in Montana. And the Hell Creek Formation actually preserves the impact boundary layer. And so one of the things that we're doing is we're working with various other research groups that study this time period, and we're collecting microvertebrate fossils from just below the boundary to get a better understanding of how the diversity of life changed in that last million years before the KPG extinction. And we're doing this by collecting sediment. We screen wash it to collect all the microvertebrate fossils and concentrate them. And then we'll be sending this out to schools around North Carolina and around the US for middle school kids to actually pick the fossils out and identify them and send those data back to the museum. And so that will become part of a big citizen science project that we're launching in the fall called Cretaceous Creatures. And in that way, those students will be contributing real data that we'll be using with our scientific team to help understand the diversity of life leading up to this extinction, so we can better understand the impact of the extinction itself.
Wow, that's really cool to have middle school students helping to collect microvertebrate fossils. And by that, I assume you mean things like really small fossils, teeth, and other really small animals. Is that right?
Yeah, so usually these are the tiny animals that live in the environment. So things like small mammals and or even juvenile or baby dinosaurs, as well as lizards and salamanders and frogs and snakes. And also birds, all the things that we don't generally think about when we talk about dinosaur ecosystems. But of course, those ecosystems include all of those types of animals, just like ours do.
Mm-hmm. Well, it's really crazy for me to think that there are all these fossils that are preserved, frogs and salamanders, the birds, the mammals, all preserved in the fossil record. What do we ever hear about? Occasionally mammals, but mostly it's like T-Rex. But all this stuff is preserved. That's really cool. All right. Well, we are at 7:30, but I think I want to take just a few more questions because they're still coming in. All right. If we can stick to the KT boundary, or I'll give you the choice, or you can talk a little bit more about what's happening in your quarry. Choice?
You're the host.
All right, sounds great. In that case, let's go back to the KT boundary. Let's keep talking about destruction. Kayla wants to know if you have any idea how long the KT extinction might have took, and if there are any kind of atmospheric similarities between then and now.
How long did probably a few thousand years. It's very, very, when you start doing geology, it's very, very hard to get that kind of temporal resolution, right? Because the methods we use for age dating rocks typically have, somewhere on the order of 100,000 year give or take. They have gotten down, this is one way the technologies have been improving in dating rocks. We do have methods now that can get you down within a 10,000 year window. So we're going back and redating a lot of these things that have been dated in the past and getting much firmer dates. But, it's hard to get any resolution below, say, 10,000 years right now. So you can say exactly how many years that extinction event lasted.
Wow, there's so much power of data in the fossil record to look at millions of years worth of time. But 10,000 years ago in North Carolina, there were woolly mammoths walking around. There were giant sloths. That's crazy.
Yeah, and I should preface that by saying that I'm talking about when we're getting into the many millions of years age dating techniques. We of course can date younger things with carbon dating, which you can use for things that are about 30,000 years old or less and get very precise younger dates. But once you get into the millions of years, the types of techniques you're using just don't have that kind of resolution.
Okay, great. Well, Madeline does want to know about the fossil site that you're in now and generally kind of some techniques that you guys use. And Madeline asks, the excavation of fossils can result in the destruction of the specimen. What are some of the measures that you take to minimize it? And what do you do if a portion breaks?
We glue it back together. Essentially, I mean, things always break in the quarry. Most of the time, the fossil is more fragile than the rock it's entombed in. So as you're chiseling out the rock, the fossil will break. Sometimes we have really hard fossil buried in really soft rock, but that's kind of a paleontologist's dream, and that's pretty rare. So what we have out here, we have consolidants. I don't have any with me right now, but they're basically in these plastic bottles, and they're resins that are dissolved in acetone. And so when we expose a bone, we'll pipette this liquid consolidant on there, and the resin will sink inside the pore spaces of the fossil, and the acetone will evaporate away, and it'll consolidate and hold that fossil together so that it doesn't break apart while we're excavating it.
Oh, that's slick. So it's like a resin coating that just encompasses the bone. So it sounds like it's different than super glue.
Well, it's very different than super glue because we work with museums, and so everything we do to a fossil, we want to be reversible. We want it to be archival. And so what's lovely about this consolidant, and I happen to have a chunk of a sauropod sitting here, so you can see it's broken because the rest of this chunk is still in the hill. But you can also see it's shiny. And that's because the resin that's holding it together right now is shiny. And what's nice about it is if you reapply acetone to it, you can completely remove everything that you put on. So it's an important tool for conservation of fossils.
Okay, wow. I'd never really realized that paleontology had to consider conservation in the museum once the specimens were out, because your fossils I thought were just rock. Okay, well, I think it is now 7:36. Yeah. So I think we might just wrap it up here because all the questions seem to be kind of diverging away a little bit, talking about modern day mass extinctions. And we have some ecologists coming up soon in the live streams that probably will be able to talk about modern day mass extinctions. So, Lindsay, I want to thank you so much for your time. And I want to thank your crew for being very well behaved behind you. And so everyone, next week, we have Naima Harris attending the live stream. She'll be talking about some large social issues dealing with ecology in both West Africa and here in North America. So please tune in for that. And Lindsay, I really wish you guys the best of luck. Stay safe. I would say stay cool, but I guess you'll do the best you can.
Yeah, we'll do the best we can. It's interesting to combine modern day climate change with dinosaur climate change in this particular quarry at this particular time.
How apropos.
Yeah, two worlds colliding. Yeah, thanks for hosting, Bucky. Good to talk to you. And thanks to the students for joining us today.
All right, everyone. Thank you so much. Bye-bye.
Bye.